Hello, everyone, and welcome to another exciting International Carnivorous Plant Society's Roundtable. Today, we are going to be talking about breeding Nepenthes, and we are joined with some great panelists today. But first, I wanted to point out that I am wearing a Nepenthes biculturata shirt with some ants on it, and you can buy this shirt in the link below. And when you buy that shirt, you are supporting the ICPS's education and conservation initiatives, which include hosting these roundtables, hosting webinars, creating animated videos, and most importantly and most fun, we provide funds for teachers every August around the world to add carnivorous plants to their classrooms. So if you want to buy this t-shirt or our mugs or our tote bags, you can be supporting our education and conservation initiatives directly. And with that, we are going to go around with the panel and they're going to introduce themselves. And then I have a couple of questions from social media as well as my own, and we're going to have a great discussion. So with that, Josh, you can take it away.
Great. Thanks, Kenny, and thanks for having me on here and for getting us all together. It's nice to see some familiar faces that I've worked with over the past decade or so. My name is Josh Brown. I run a company called Predatory Plants out of Half Moon Bay, California. We are a kind of full-line carnivorous plant online retail nursery with most of the genuses at any given time being represented, but I do specialize in Nepenthes breeding and hybridization, particularly over the past 5 years or so. It's become a pretty serious part of my program, and I have a brick and mortar store in Half Moon Bay as well, and I'm starting to scale up with more national wholesale kind of operations going on. So pretty exciting times for Nepenthes breeding in general.
Yes, I'm up. So I'm Drew Martinez. I'm the owner of Carnivero. We're based out of Austin, Texas, and we are a carnivorous plant e-commerce nursery. We do a lot of the carnivorous plant genera, also some tropicals such as aeroids, but Nepenthes really our bread and butter. And we focus on a wide range of breeding, everything from lowland to ultra highland Nepenthes. And I'm really excited for where the market's going now. I think with all this excitement coming for plants in general since the pandemic hit and a lot of people getting more into house plants, I'm really excited to see where Nepenthes are going and how the industry is growing.
I'm Rob Cantley, Borneo Exotics, which is a wholesale nursery based in Sri Lanka. We grow specifically Nepenthes and have been breeding them here for about 25, 26 years. As other people have mentioned, it's an exciting field. It's growing fast. Since the pandemic, there's been a lot more interest in plants in general. So we make a lot of hybrids. I think probably 2 or 3 a week, and have done for a very long time. So it's a long, continuous process for us. And this is my wife, Diana Cantley.
Hello, everybody. And it's a pleasure to be with you all. And I've been in the business with Borneo Exotics for 23 years now. Quite exciting, particularly when we had the lowland nurseries and the highland nurseries, because then we had the best of both worlds to be able to hybridize between the lowland and montane species.
All right, very good. And we're going to start with that question, Rob and Diana, and I'm going to ask everyone on the panel. What is your breeding goals? What traits, what characteristics are you looking for when you breed Nepenthes, other than you have 2 flowers? So Rob and Diana, you want to start?
Yes, of course, you have to have 2 flowers, male and female, which if you don't know whether you've got male or female for quite some time after you first start growing the plant. Traits, the obvious ones that people would talk about, I think, would be physical size. We just happen to be sitting in front of a fairly large one here. The nature of the peristome, whether it's toothy or broad. also, not to dilute particular characteristics, if you cross a very toothy peristoned plant with one that has no teeth, you generally don't get something that's all that interesting on the peristome front. But there are other less obvious traits which people may not think about when they're breeding, but the toughness of the pitcher, like this is a Cebuyanensis cross, and the pitcher is very tough. And also how easy it is to pitcher, how many pitchers it produces. You know, sometimes the plant will only produce one pitcher every 3 or 4 leaves, and that's not much good commercially. And then we also choose for certain traits that are wanted by mass producers like the Dutch. They have different criteria altogether to hobbyists who are generally more patient with the plants.
But we like to choose, I look at plants that have particular characteristics such as producing a lot of pitchers. They may have soft pitcher, but then you cross it with something which is mounted species, for example, and then you get something which is much tougher. So then you hope that you're going to get something which has a dynamic look to it, but it also produces a lot of pitchers.
Drew, what is your motto for breeding Nepenthes?
Well, it depends on who I am breeding it for, right? So we get everything from the most picky, highland species that are dentate. You know, for example, Nepenthes macrophylla, Villosa edwardsiana hamada. And, essentially we don't want to dilute those characteristics. So we go for even more toothy characteristics, say, right? And that would be for the collectors who are capable of growing them. Which they are a challenge, right? If I'm breeding for something that's more vigorous, then often hybrid vigor will be targeted. So something like from a montane species crossed with a lowland species could be a good combination. It's it'd be targeted to grow in a wide range of conditions. And then there's also some plants that I might be targeting for just color and shape of peristome, such as Nepenthes vichii or Nepenthes truncata crosses. And if I'm breeding for collectors, I'm trying to go for the most superlative characteristics. But if I'm breeding for something that's going to be more palatable to the everyday houseplant person, then maybe it's something that's just easy to grow. Something like ventricoca crosses or some of the other more vigorous Philippine species, crossed with something that's really showy.
Josh, what about you?
Yeah, it's been a serious evolution actually for me. Like I said, I kind of been like a lot of folks who have extensive Nepenthes collections, a backyard breeder sort of, like I probably pollinated my first flowers in 2014, 2015. But then really focused on it starting around 2018 and producing hundreds of crosses a year, experimenting, seeing how they all work. So you kind of slide from opportunistic breeding, which is the excitement of like, I have 2 flowers and you just, doesn't matter. Like we'll just do it, see what comes out, to more and more refined kind of strategies. So I've gone through the collector phase that Drew was talking about, where people who grow Nepenthes like toothy stuff, broad stuff, colorful stuff, which is a kind of moving target where things become more popular as they become more visible in the marketplace and more coveted and that sort of thing. So I've done a lot of work on making the crosses that I would want as a buyer, and that informed a lot of my early decisions. But in my most recent breeding and more experimenting on the things that I'd produced, it's becoming more and more clear that, like Rob said, you do have to take into consideration stuff that the collectors might not be that interested in even hearing about. But he cited the Dutch, which is a great example because they have a much more advanced horticulture industry in the Netherlands and in Europe. Because of that, things can just move through the EU. So their Nepenthes market, along with all of their other horticulture markets, is just much more advanced, and it's kind of if you look at it, it's where we're going to be in 5 years, usually across the board. They're just that much further ahead. And like Rob said, they look for different stuff. And as a grower, a lot of it has to do with, or as a hybridizer, as a breeder, it has to do with what the growers are able to do. So I don't know the Netherlands especially well, but in America, all of our plants that anyone buys at any store are grown in 2 places, and it's Florida and Southern California, basically. And that's just thousands of nurseries there, whole citywide greenhouse complexes. And if your plants don't grow there, you're kind of stuck growing your own produce, and you can't really sell them to other growers and move them around the country through anything other than a small online presence like I have, or local brick and mortar, which I've also developed. But moving into that next step, a lot of it is, does this grow in our exact climates where we grow these other plants? Rob said toughness of pitchers. That's cited all the time because they get damaged and no one wants to buy them if they're floppy. The Sibionensis hybrid behind him has tough pitchers, but also has 3-foot long attachments to the tendrils. That's impossible to ship, we have learned. And everyone wants really short attachments, which is why I think they've been focusing at Borneo Exotics on Ampullaria hybrids, because they have microscopic attachments and then a nice big pitcher like their Lady Luck. And that's like the Dutch ideal right now. Color, but also the ability to move through a pre-existing system of how these plants make it to market is my new big focus. I'm not doing that much hybridization right now because I have too many crosses to kind of work through, but the plants that we're selecting are really focusing on, like, is this marketable? Not as much about, does this have enough Edwardsiana in it? Like it has these traits that make it easy to move through the existing horticulture systems that are in place.
So Josh, what is your philosophy on like a Nothospeces, where you're just breeding like one species and another species, or like super complex hybrids?
Right. So it varies. It's not an either or, and it's not necessarily consistent. The general trend is that the more complex a hybrid is, the more variable the offspring are. And if you're breeding for consistency, you really that adds a lot of extra work to get to the plant that you want from an ultra complex hybrid versus, most primary hybrids where you just take 2 species, their offspring are much more consistent. And likewise, there seems to be kind of some trends. I'd love to hear what other people who breed Nepenthes have seen from this, but there are also compatibility issues within like the complexes of Nepenthes, like the Maxima complex. If you're breeding within that complex, it's even more stable versus you introduce something crazy into that. Sometimes, like from an Inermis or an Aristolochioides, like these weird orphan species that kind of have very strange traits and are genetically isolated in some of the cases. We have seen just strange outcomes from that, either not very viable crosses, or can't really figure out where they want to grow in the greenhouse, just because they just weren't, that was never supposed to happen. So sticking with highly compatible, proven, we're having a lot of success with back crosses, which is like you do Veitchii Maxima, back onto Veitchii, back onto Maxima, and you get really stable, really vigorous lines out of that. And so that's, my philosophy hasn't developed yet, as you were asking about, but we are developing philosophy and praxis and like how we get this stuff done in our breeding program. And that's now informing what pollen we put on what flowers, now that we've grown out 500 crosses, seen what they do, seen how they perform in tissue culture. That's another one that like has to really, if it just can't thrive in vitro, then you're just doing all vegetative propagation, and like what you just sell them and get rid of them. So that's another big piece of information that we get before we even see what a pitcher looks like, is like, did this grow well in vitro, in tissue, for the audience, in tissue culture. That's how we germinate our seeds. And if you want to sow on top of soil, that's its own industry. But if they don't thrive in vitro, generally we kind of just plant those out and sell them off because we don't really have a plan for them beyond that.
Drew, what about you? Do you like
Yeah.
the complex hybrids?
Yeah. So, let's see. I mean, I breed some really complex stuff, right? We're to the point where like it doesn't fit on a tag anymore, right? There's like 12 parents plus involved, right? And you know, I think it's most important to choose once you get that complex, you're choosing the superlative characteristics of the complex parents. You know, it's not just I don't know, lowii vici boschiana cross with vici cross with something else. It's like you got to pick your best ones to really bring out the characteristics that you're looking for. Otherwise, they just kind of get muddled out. You know, in terms of complex hybridization. You know, well, I'll say this about hybridization, right? So I think, like, at least when we're talking about a collector market, right, and then I'll go over to, like, you know, what we were talking about before, the mass market. Like, I think there's been an evolution over the last 15 to 20 years where there was a while where basically any Nepenthes hybrid was novel to the community, right? Where like you could have a ventricoca cross McFarlane, and a lot of people would be interested in buy that because that was a novel thing, right? And now I think we're getting to the point where that's not quite novel. Like it's, you know, it's not worth my space anymore. Like making hybrids that are not going to be novel or interesting to people in the either who are in the hobby as collectors or plants that have like mass market potential, right? So in terms of mass market potential, I think Josh mentioned something about sowing things, you know, in vitro versus soil. We are actually taking an approach where we're investing a lot of research into reintroducing Nepenthes into tissue culture from meristem. And that is one way where we're getting around like this genetic diversity where, you know, instead of having to sow, you know, 200 seeds and flasks out of a cross and just, it's a crapshoot, like what characteristics those 200, you know, are going to have. And you have to raise all 200 of them, right? And I mean, I'm sure Rob can talk more about this because they've actually done it, right? Like we're in our infancy kind of doing this stuff, but they've actually done this over at Borneo Exotics to select cultivars, right? It's a lot of work. So like we're trying to take this reverse approach where we can grow a whole bunch from seed because that's pretty easy. It's like low, you know, labor, low maintenance, and we can find the 1 out of 200, you know, in that batch that has those supportive characteristics that we want, and then get that back into the lab, right? And it's not an easy thing, but it's also not as impossible as it's been made out to be. You know, at least in the, you know, in the hobby so far.
And that's great news.
Yes,
we're all very happy to hear that someone's doing that. And it's
yeah. I mean, it's been done once, right? Like it has been done.
Sure.
Actually, Nepenthes Gaia. That is, I think it might be the only one where Sam Estes bred Nepenthes Gaia. This was back when he was in his prime. You know, I think it was 10 to 15 years ago. And he actually produced some plants, you know, for some tissue culture labs, you know, from cuttings with the attempt of, you know, them getting that into in vitro. And they were successful, right? And Nepenthes Gaia, that's a great plant, right? You know, I would say, you know, in terms of like actual, you know, plants that have potential for the mass market, like that's one of like less than 10, you know, out there, right? And, you know, I think Rob and Diana produced most of all the rest of them, right?
All right, so Rob and Diana, I feel like I know your catalog pretty well, and I don't know how many hybrids have more than 3 names on the tag. So can you talk about your idea about breeding, hybridizing?
Firstly, a lot of what Josh and Drew have said really resonates with me, particularly the story about how Josh in the early days would cross anything with anything. It's exactly what we did. Literally, something flowered, you'd cross it, and you're making mirabilis crosses and various crosses which have really got little value. So as the years go by, you do get more selective. And you're talking about complex crosses. You do get this enormous variation, as Drew said, and I think Josh mentioned, a massive variation in the output, which means you can't really predict as you can with a simple cross, say Ampullaria cross, take an obvious example, Rafflesiana, you're going to get something that looks like Hookeriana. You've got a pretty good idea what the outcome will be. If you're making complex crosses and you're putting them in the lab, then in the early days, we would actually keep a lot of clones. Drew knows this, he's been here. These days, we've got 2,500 crosses and we can't keep that many different clones in the lab. So it is a crapshoot, as you put it, because we basically just have to pick a few clones and hope they work, or we mix them, which means we get a very variable output. And some people like that because you can get different forms and colors within the same item. And some people don't like it because they're unlucky and they don't get that striped peristome they wanted from the Hamata Velosa cross or whatever it might be. Mm-hmm. So there's all sorts of different arguments, and without a massive laboratory and infinite capabilities, we are getting very, very selective about our breeding as well. We get a list of things that flower from every week, male and female, and we sit there and we decide what we're going to cross with what. And there's lots of crosses now that we would have made years ago. We don't make anymore. Before I pass it over to Diana, I just say, Drew, it's fascinating about the meristemming. I've got to talk to you about this. This is the holy grail if you can get that to work in any way reliably. But you know, if you have a very fantastic rare plant, you don't necessarily want to risk killing it by taking off all the meristems and failing. But if you've got a sufficient supply of plants and it can be done, that is really good news. Mm-hmm. Diana, you got anything to add?
Yes. Well, we were just talking about the variety that you can get, and certain special characteristics. Sometimes, of course, we've done hundreds of, well, we call them clones, but they're not specific clones. As you know, we take one seed and then that gets multiplied up and we give that particular number so that we know what particular characteristics that has. And then ultimately, years later, maybe 2 years or maybe 7 years later, depending upon how fast they grow, we can discover which the best is for production. So it's, as you all know, it's a very, very long, tedious process, but in the end you get good results, hopefully. Now this is something I've just brought from our clone selection nurseries, and this is the same hybrid, but as you can see, you've got a very, very different colorations coming out of it. So it's a very interesting process, as you all know, but it is time consuming. And as Robert was just talking about, when you get more complex hybrids, then it really is potluck as to what you're going to get yourselves. But I have had to go through, as Drew knows, with thousands of different hybrids and just say, look, this doesn't work. This is not our best hybrid. We've got to just get rid of them. And then, of course, Rob comes along and somebody has asked, quite often it's somebody, the Dutch, oh, we've seen this plant in so-and-so's nursery. Can we have it? Well, I'm sorry, but it wasn't one of our best.
That hasn't happened for a week now.
so it's gone. But yes, so I have to be very very strict with what we actually maintain.
Yeah, Diana has the power of life and death. I started hybridizing when I was 20, and look at me now. So it's a long evolutionary process.
All right, in a minute, I want to ask everyone about how they decide what to keep and what to cull. But first, I want to ask for the listener, the actual mechanics. We're going to assume that the listener can identify if the flower is male or female. And then Robin, Diana, can you say how you collect the pollen, or do you store the pollen for any duration before you fertilize the plant? I'm thinking maybe if a listener has a male that blooms and then they have to wait for their other plant to bloom female. So Rob and Diana, do you want to talk about you moving the pollen, storing the pollen?
Yeah, we tend to do it, Josh, in real time as much as we can. We get much better results if we have things flowering at the same time, which is why we get a list every week from the staff about which plants are in flower, because you're almost guaranteed a success with that. If they're not flowering at the same time, there are methods of storing pollen, and I think probably Drew and Josh would be much better to explain the methods for doing that. We rarely do it, but you have to keep it very dry. You have to keep it cool, and you just don't get the same results, but it can work. But often when people send us pollen through the post, we find it's just not viable, even though they have done their best to store it properly. It can be disappointing. So we've often then used one of our best females with a pollen that somebody sent us and don't get anything from it. So, for us, it's a little bit difficult. Maybe something happens to it in the post, we're not sure. So I haven't really got much else to tell you about how we pollinate, other than it's as much as possible in real time. And if we store pollen, it's for a limited time.
And we do keep our male and our females in completely different nurseries, because we have in the distant past, discovered that something has pollinated without our
permission.
Yeah. Some virgin births.
Disastrous. There's no hanky panky allowed anymore here around here. You find out years later that it's not what you wanted it to be, and that's a very long, expensive process.
So it's very, very important to actually recognize whether it's a male or a female before they start opening, and ensure that they're kept well apart.
Josh, does Predatory Plants have a whole video about pollen and storage?
Yeah. So, well, we have one about specifically pollination. So on our YouTube page, it's pretty well trafficked. It's the ultimate Nepenthes seed guide, something like that. We made it in 2017, 2018, but it still applies. Like Rob said, 99% of our breeding is we have them in flower at the same time. We actually generally remove the whole anther, which is the structure with the ball on the end that has pollen on the ball. We have that in forceps. We cut it off and we take it to the female and literally touch the anther to the receptive surface of the stigma, and sometimes use like a flat-headed toothpick to spread it across even more evenly to get a better pollination event. If there's a bit of a lag there between when the female is going to open and when the male is shedding pollen, I will collect pollen occasionally. In a similar vein, I remove the anther, and then I have little vials for centrifuges, is what I typically use, and take a X-Acto knife and look very closely and scrape the pollen into the centrifuge tube, because I think, since we said for the collector, what should they do? The biggest mistake people make is they say this anther is the pollen, and they just remove the plant structure and they wrap it in foil or something like that, and that introduces a bunch of junk, a bunch of water, a bunch of problems. And so we will, if someone begs us to get some pollen from them, a lot of times we'll just get a mucky envelope with some withered anthers in them and don't even know where the pollen is in there. And so the big thing is to remove the pollen from the anther completely. None of that structure matters. It's not going to get you anything else. So completely take off the pollen at the very least. Then Drew has actually much more experience on storing pollen than I do, with drying with silica, with deep freeze, like that kind of thing. If I'm storing pollen, I am mentally counting the weeks, and basically once it's a month or two old, I'm not wasting flowers on that pollen anymore. I keep it in the fridge. I keep it in a sealed container with no moisture. But without a bit more intervention than that, the viability just goes like that. And we also are privileged to have a big enough collection that usually enough stuff is in flower that I don't need to be trading for pollen. I don't need to be storing pollen. So we just make the hybrids that we can and trust that if there's something we want to make, sometime they'll line up and it'll work out pretty well. But that's just a nice to have. But focusing on really high quality pollination events, which is using the freshest pollen on the best flower, is way better than making some long shot hybrid that you're dreaming about, at least for a high volume company, like where we're trying to just make plants and see how they go. And we have the greenhouse space to grow them out. It's more about just really optimizing that time and those flower pods and your tissue culture and your employees' time when they're potting things out. Like that's our that's our big focus. Is just like we want really good seed, and that's only achieved by using really fresh pollen. So storing, not yet.
Yeah, we we do store pollen, but like Josh said, you got 1 or 2 months where you can have any confidence at all, and beyond that, it's a crapshoot. You know, like I'll throw stuff in the fridge. I'll like be really disciplined, and you know, like we have a process, right, that we've designed around, you know, just basic desiccant and, you know, keeping it slightly warm and to dry it out, and then, you know, storing it cool. But, you know, past 1 or 2 months, kind of like Josh's situation, we have so many things in flower that it's just better to do something that's going to be reliable than to, you know, to risk pollen not working, right? I mean, in some cases, I've stretched it out as long as, you know, 4 or 6 months using pollen in the freezer, but that was more for like things that were conservation focused. That like, for example, like I exchanged pollen with University of California, Berkeley, right? They in the greenhouse they had a female clipiata, and you know, for conservation purposes, that's huge because that's one that's quite in peril. And they asked me, "Do you have any pollen?" Well, I happen to have some in the freezer at that time. And I sent it, and we didn't get great results though, right? I got a few that came up in the lab, so we're multiplying that out. You know, I don't know if they got any germination out of their stuff, right? It just, you know, but I think that was a worthwhile endeavor because, you know, that's a species in peril.
All right, now this is my favorite question. I've done a couple of these roundtables. This is the first for breeding Nepenthes, but I've done breeding Venus flytrap so far and breeding Sarracenia. And the question is, how do you determine which ones you're going to keep and which ones you're going to cull? And at what age do you determine this? So Rob and Diana, you can go first. And I'm in my mindset, I'm thinking you have a cross, you get 100 seeds, and then Rob and Diana, what do you do?
But if it's only 100 seeds, we're quite disappointed to start with. But no, sometimes it is, and if we get a small number of seeds like that, we'll put them all in the lab. And if it's a cross we've been striving to make, we might even sow them individually, so we keep the clones separate. That's the extreme. The other one is if it's cross we made before or we're not that excited about, we might just mix them together and what we call combine them. If we get a large number of seeds, we'll do that in the lab with some of the seeds and the balance go into the nurseries in the traditional way to be sown on and for seed grown. We don't waste anything. But really, I'll pass over to Diana because I drive a computer these days and she really runs the breeding programs here. So she can tell you when she has the power of life and death and decides to cull things, the criteria. Yeah.
Well, the culling process, really, you know, you're talking about at what length of time. Well, it very much depends upon the speed and rate of growth of the individual hybrid, or whether it's a species. With the species, of course, we're getting seed that even if we have collected the seed, you have no knowledge of whether it's a true species or whether it's being hybridized in its natural environment. So we have to grow them out for sometimes many years before you can identify and ensure that what you are producing is the true species. So there's that side of it. And of course, many of the species are a lot slower, as we all know, than our hybrids. Hopefully the hybrids are going to be a lot faster. And we are actually sitting in our specimen nursery now, and many of the plants here are 20 odd years old. And so I go through and I just compare, particularly the hybrids. I mean, species we keep because they're the holy grail. some of which I would say aren't really worth keeping, but then that's me.
Often for conservation reasons,
Yes,
more than commercial reasons.
Exactly.
There's only 100 of
Yeah,
them.
yeah. So, but it is, I mean, well, that's another subject, isn't it? That people are finding new species every year, but are they new species or are they actually hybrids? But we won't go there at the moment. But yes, so for the sort of the process of saying, okay, I like this, and the other one, it's whether it is really got fantastic traits. Is it an attractive shape? Is it an attractive coloration? Has it got the peristomes that we were really looking for in that hybrid? Because you know, you hybridize something with petiolata, for example, or and you want that coloration, you want that gorgeous pink coloration from the petiolata. Or if I'm doing something with a red leaf, that's what I want. I don't want a green leaf to come out of it. I want that red leaf going into the peltata, for example. So some of them work and some of them just don't, and you just have to say, okay. And some of the hybrids with toothy, as Drew was talking about, you know, you're looking for that trait to come through, and it doesn't always happen.
Yeah, you
have to get rid of them.
I might add also, as Josh mentioned, it depends on who you're breeding it for as well, because if it's for the Dutch, they require the short tendrils. So you're selecting for that particular characteristic, which isn't necessarily that interesting for the hobbyists who don't mind the 3 foot long tendril, as long as a picture on the end of it is what they want to see. So you've got to think about your market as well when you're making these selections. The one that we alluded to earlier, where we were told off basically by a Dutch customer who wanted something that we destroyed only recently, it was just, it happened to have the traits that he wanted. And when we were actually looking at it and viewing it and deciding whether to keep it or not, we weren't considering the Dutch at that particular moment. I think we might not have destroyed it had we done so.
Well, also what he didn't realize is that it had glandulifera in it. And of course the glands make this very, very difficult for mass production anyway. It might have looked good in a particular nursery where it's a specialist grower, but you've got this tendency for the glands to have the black sooty mold, and so for a production plant, I don't think it's a goer, really.
Yeah, they go for speed of growth as well, and all sorts of characteristics are not necessarily that important to the discerning hobbyist.
Yeah.
Diana, if you're looking for like a striped peristome and you have 300 seedlings, the juvenile pictures might not show the adult characteristics. So can you give me a guess, or the audience a guess of, like, do you get rid of half of them after 2 years? Or are you talking to the other people in the nursery and saying, do you like this? Do you like this? You know?
No, I don't talk to other people in the nursery.
Heard this.
No, they're great and they do a fabulous job. They really do. Well, there is one guy who does point out things to me that he thinks, this is looking really interesting. But generally, as you say, actually at a small size, you can see that there might be something which is going to be more interesting than something else. But you do have to wait for that process of developing the picture.
Yeah,
probably on an average 2-3 years. Yeah.
If it's a Vicii cross, for example, you're looking for a stripy peristome, it doesn't necessarily become evident for quite a long time. So you can't, if we're doing what we call clone selection, and we have a whole nursery full of thousands of plants where we're looking at clone selection, we have plants that Diana just showed you an example of. They're barely big enough yet to actually make that judgment, and they've been there for a couple of years in some cases.
Oh yeah.
Yeah.
All right, Josh and Drew, same question.
Sure. So there's actually a little bit more to expose here, because at least the way I think you're asking it, we're not there yet. If you're talking about selecting cultivars, like individuals, we're still, like I said, how far behind we are other markets. Like we're definitely the biggest breeders in America, but we're in the process right now of selecting lines and crosses. So we're growing out hundreds of these crossing events that I did over the years, and just figuring out which lines are doing well, which populations are doing well. Again, from all the way from the tissue culture to the planting out, the hardening off, and then how fast they grow out, and then what the traits are. And then if there's a standout individual in there, like, what do we do about that? We have no ability to be looking forward to that right now, as we just try to figure out what our line of crosses are. Like we're nowhere close to doing that next step, and we don't even necessarily plan on doing that. If we get stable populations from lines that breed true, that have the characteristics we're looking for, we don't necessarily need to select down to a specific genetic individual if we can continuously produce it. There's plenty of agribusiness that operates that way, where they just operate from parent stock rather than a single individual that will be replicated over and over. That's not the case for Nepenthes specifically in the market. Every mass market Nepenthes that I'm aware of is a clone that was selected arduously to get to that individual. But we're still focusing more on what crosses work well, what can we replicate. If they're like with high variation crosses, we can start saying like the red ones from that cross are very cool, so we should keep an eye on those. Or that one makes some like 20% have remarkable stripes, so let's just make a note of that. But the process of then getting that to market is just several more years than we currently have done. And we're really happy to be producing just good lines of plants that can enter the market, both at the collector, the retail, the brick and mortar, the wholesale kind of level, to where like getting the next blockbuster cultivar is pretty far from our mind right now while we do the rest of that process.
So if there's 20% of the seedlings that you enjoy or you think they have potential, are you culling the 80%? Are you selling the 80% at a farmer's market?
The cool part about the way I happen to have structured the business is that it's vertically integrated. So that means we have representation at all the links along the chain, right? So we're a breeder, so we produce the primary genetics. We have contracts with labs. The labs send us. We are a hardening nursery that brings plants out of culture into active growth. We can sell those young plants to other nurseries to grow out. We can grow them out ourselves and sell them online through our various online platforms. We have a store where we can sell to people locally. We have local small like nurseries and businesses that have plants that come by and buy stuff. So as long as we have the outlets, we're not like binning plants.
Mm-hmm.
That's something that I still don't know if I'm emotionally prepared to do. But it's kind of inevitable when like with this kind of thing, it is exponential. Like we have like 20,000 plants on the bench or something insane like that right now. It's growing fast. And so if you're not constantly feeding the beast with new sales, you run out of space or you just throw stuff away. But so we don't have it's not necessary right now. We are like preparing for what the answer to that is. Like, when do we throw stuff away? Like these benches cost a lot of money and someone has to water all of these and someone has to make sure they don't get pests and stuff. So that just like picking cultivars is kind of right on the horizon. We're like, wow, we're getting pretty big. We have to do stuff like this, but we're not in a position to be like throwing away a bunch of plants because they don't have stripes. We sell them to people who don't want plants with stripes, right?
Yeah.
So
for the 20% or in the future, the top 10%, will you replicate that breeding, or are you going to put those individuals in tissue culture?
Yeah, no, we're going to go straight to Drew with his new magic process, and he's going to just get them right into culture for us. And it's going to be super. Like, that's it. That's all I have to do. And
Drew, you'll also be getting plants from Rob, it looks like.
yeah, no, we're all just gonna send stuff to Drew and be like, just deal with this, and we'll see you in 3 years, and just like
Yeah.
dump a bunch of cultures on me, and we're
Yeah.
gonna be fine. But no, the answer is like what this has been alluded to. The way this works depends on how you sowed the seed. So if you sowed a bunch of seeds into one petri dish, they are combined, and you can maybe pull lines out of there. If you do like one seed, or you do them on a grid, you can like focus on individuals. But the only way, unless it's a percentage play, like you said, like, oh, 20% do well, so we have to take 10 lines out of this culture, 20 lines, 30 lines, to make sure we get at least one good one, then replicate those purely, grow those out, find the ones with the traits. This is the stuff that I'm not doing, is the answer to your question. It's like, is the
No,
whole
you'll end up
process.
looking like me if you do.
Yeah.
Yeah, it's just like, okay, see you in 25 years, and then we'll be like,
Yeah, that's it.
No kidding.
yeah, but that's why our businesses are at different points in our product development cycle, right? Like, so Rob can be looking at me like, oh yeah, I remember when I was doing exactly that process, and then we refined to this, and then we introduced this new step, and then we had the luxury of doing that, and then so that's what we're ramping up into right now. Now that we have at least the first step of a breeding program, of a tissue culture program, of a way to get them into the marketplace, so like a well-developed e-commerce solution, and starting to build these wholesale relationships. That's the only way that you can support all of this process that takes years and years and years to bear fruit. You have to be doing something during all of that time. You have to be selling something. You have to have a product line, and so that's what we have now. And then once that, we settle in and we stabilize on that, then we can start talking about like, okay, this one plant though is really good. Like, how do we make 1,000,000 of these instead of 10,000? Like, who's going to do that for us? Where are we going to grow them? So that's what we're looking at. But right now it's just about, like they said, waiting a couple years,
Yeah.
see how these look, wait a couple more years, see how they look again. So it's not like impending doom, but it does ramp up over time, and it becomes more and more of a refined process.
In the ICPS roundtable for breeding Venus flytraps, we had Matza from Green Jaws, and everybody was, drooling over his cultivars. And he goes, well, I crossed those 5 years ago, 7 years ago. And he doesn't do tissue culture. And he's like, I grew them out. And then I selected, and then I had to wait, years to figure out which are the keepers.
Yeah.
All
So
right.
basically, between Venus flytraps and Nepenthes, you just have to add an extra like 15 years.
Yeah.
And the Venus flytrap, if you like one, you wait for it to flower, which happens year 3, 4, 5, somewhere in there. You cut off the spike, you put it in culture, you have it. Congratulations, it's in replication. 8 months later, you can be planting them out. Like, it's crazy how easy that particular role is compared to like ornamental tropicals like these ones. But even that's like you said, oh, that's 7 years, like crazy. This is 20 years. And so just by the nature of the plant, until Drew gets his process going, that would knock, that's why Rob and I are kind of salivating. Is it like, wow, that saves like a bunch of time if you just do that.
I mean, that's the focus, right? So like, we've got a team of biologists. I love my lab staff. They're skilled, and honestly, Austin is not the cheapest place to live, and pay employees and all that stuff, right? So like, we are highly skilled, so we can focus on research projects like this. But in terms of like culling things and the greenhouses, we kind of let nature play out. How it's going to play out. So we've got a high quality standard for all our products, and if it doesn't meet that standard, it's not leaving, right? And at some point, like if it's just a run for too long, like it's getting tossed. But like whenever we sell, we have found, it all depends on the market, right? We found collectors are perfectly fine taking extra small and small plants, right? Like, and that's actually great, because I don't really know what their exact characteristics are going to be in that size, right? Like, so we choose our winners based on, our winners, right? Quote unquote, based on vigor. But I don't know if those are going to be the most attractive plants that collectors are looking for in the batch. So like I honestly don't know, like whether it's random selections, tissue culture plants, or seed grown plants. like whenever you get a small plant from us, like it truly has potential to be better than anything we have in the nurseries, right? And I think that's appealing for, if you're a collector, right? Like it's like it's exciting, I guess. Everybody likes to play the lotto, even though like the lotto is very low odds you're gonna win, but everybody plays, right? The lotto's been around forever, right?
Yeah,
So yeah,
you have one good one out of Josh's 20,000. You could be
Good
the
odds.
winner.
Good odds compared to the
yeah.
lotto.
Mhm.
Yeah, that's right.
Yeah, that's pretty
Okay,
good.
now speaking of lottery odds,
Mhm.
can the four of you, can you talk about variegated Nepenthes? Is it environment? Is it genes? Is it stable?
Yeah, so I was just
Discuss.
talking about this, and I would like to know this because so
Mhm.
I started my program again. Let's talk 2018-ish from seed that had been like pollinated 2016, 17. So we're starting to see individuals 2018, that kind of area. And I specifically remember we sowed a bunch of seeds from my first few crosses. We plugged them all out. It was amazing. And we did get some variegated plants. And at the time, I remember calculating that it was pretty stable in our first few crosses at, it was one in about 2000, I think it was. And so that seemed
With lots of different
realistic.
parents.
Lots of different parents, lots of different situations, lots of different, there's several kinds of variegation
Yep.
that we can talk about. There's like bicolor, tricolor. There's this cool kind of webbing that Nepenthes sometimes do. But that's when I was top sowing, right? And that lasted for a few years. And then I moved into sowing in vitro. And I have produced an order of magnitude more, at least maybe 2 orders of magnitude more Nepenthes from in vitro than I did from top sowing. And I have so few variegates, it's really not worth talking about compared to the 1 in 2000, which does, at least like anecdotally for my answer, something's going on. And it could be selection bias where like variegates tend to be less vigorous and they might not survive the in vitro process of sterilizing the seed, of maybe getting outcompeted in the flask. But once we get a culture which hasn't been selected, right? So our labs aren't culling the seedlings. They sow the seed, they deliver them as they reach size, end of story. When we receive those and we plug them out, we have statistically 0 losses. We're not losing 10, 20%. And so the bottleneck isn't there where the variegates might be falling off. And so I have myself been wondering, and it's good that you asked this, like, is whatever environmentally potentially caused the variegation when I top sowed, is that not happening in culture? Is culture selecting against it? Are they getting an early like mega dose of vitamins and nutrients that's like knocking that out of their genome? Like, I have no idea, but our lack of variegation has been striking since we moved to in vitro sowing, and statistically like very striking, with big enough numbers for it to be like something that I've noticed and have them off the cuff to talk about right here. Is that like, I don't see them anymore where I used to see enough of them that like I have a small collection of variegated plants from when I top sowed, and I don't have that now from when I'm doing tens of thousands of plants in vitro. So like, conversation starter, like anyone else have anything along that? I don't know.
You guys have more statistics than anybody, Rob and Diana. I'd like, just anecdotally, I will say this: like we've got the luxury of having our lab on site, and I can peer in there from time to time. I'm not quite like you, Rob, where I'm on top of the hill. Much more than I think you'd like to admit, right? Not able to go out in the greenhouses as much. So I'm getting there. But when I peek in the lab, we do have quite a few variegated Nepenthes in culture. They're not fast though, right? They're definitely slower. So Josh, it might be like
They might just not
there may be a bias there, right?
They might not be getting delivered to me, right? Like they might not be making it to spec. And like, I just have a bunch of flasks of variegated plants that they think are just bad in my lab. So I need to like take a look. But that's certainly one of the things that I was humoring. Like, maybe they just don't grow very well in culture, and they grew okay enough in soil to make it through my system. So that might be it too.
Yeah.
Oh,
Rob and Diana, how do you guys?
Yeah.
How's that end up?
Yeah. Well, from my knowledge, anyway, I don't believe we've actually got any in the tissue culture in the lab.
We do.
We've got one.
Yeah. We didn't know we were going to talk about variegated plants. Personally, I don't like variegated plants that much. It doesn't do much for me. But while you were talking, we found one behind us here in the nurseries, which is basically a variegated Cebuensis. If I try and show you.
Drew made one of those.
Yeah, and I just looked at the label, and it is actually a tissue culture clone, but
Diana's she's
she's
iffy.
I'm doing something wrong here. It's 2653 clone 19, which tells me it came from the lab. It's a Cebuensis. It's not doing brilliantly at all. Its variegation is becoming more pronounced. It's stopped pitchering. It's got one pitcher on it.
But then you can. We have had Cebuensis in particular that have had glorious leaf variegation, and the pictures are a gorgeous shade of pale pink. So the two combined is a very attractive plant.
They're not stable.
But we tend to do cuttings, and then we have to grow the cuttings on to make sure, as Rob was just saying, that they are going to continue and be stable before we can get it out there into the market.
With this particular clone, although it's in the lab, we've got a variegated one right here. The other clones, the other plants with the same clone coming out of the lab are not variegated.
Right. So that's
otherwise, we'd have them all over the place.
Yeah.
So it's a sport. Yeah. It's developed it since it came out of the lab. Some reason. But this thing is actually not going to survive. It's no longer producing any chlorophyll. The leaves are completely white, so it's not really something at all attractive. As I said, it did come out of the lab originally. I can tell from the label, but the other clone 19s of this item, which we must have around the nurseries, are not variegated.
So I'm in Florida, which has many tissue culture labs. And I don't know how often it is, but I am familiar with their mass producing St. Gaia, Ventrada, and
Miranda.
Well, I don't know if Miranda's come in variegated, but in these flats of 72, they have 10,000, 20,000 a season. They do get multiple plants that have variegation that last for at least 3 years in the plant. So
and so that's just a stable clone that does that.
But
that's interesting to know.
But if they're just mass producing and it's supposed to be genetically identical, it's like how did those genes get turned on or turned off?
Well, it might not even be genes, like genes in a sense, right? But like there's different forms of variegation, right? I don't know, I've ridden this aeroid wave like during the craze, right? So it's been quite successful. So I've gotten a lot of variegation, a lot of variegated plants. There's chimeric variegation, which is not particularly stable because it's two cells trying to outcompete each other, and eventually one will win more than likely over time. So it's either going to look like that Cebuensis right there that's starting to peter out because it doesn't have any chlorophyll, or it might revert, but then it might come back later on. You might cut it and it will come back. There's other forms of variegation that might be caused by viruses, where traditionally, like tulip craze in the Netherlands, like historically was caused by a virus. It created some beautiful flowers, but then genetic variegation is probably the most stable. You have things like Monstera constellation, which that one breeds true actually. Like I know some people who have self that and got variegated seedlings very similar to its parents, right? So it could come in it can come in any of those forms, right? And obviously the genetic one is the one you want as a plant breeder, plant producer, because it's stable, right? Like I always get worried, variegated plants are quite expensive at least in the aeroid world, right? And you buy one and just to take that risk that it's going to revert on you. it's, it's quite a risk.
Yes.
We have,
Ju-
like Drew said about like breeding true, the most stable variegated Nepenthes that I think anyone's aware of is the variegated, it was called variegated Leda. It's probably Graciela flora. Graciela flora, fine.
Yeah.
Yeah. It doesn't have hair. It has a wider neck. Yeah, I know it's Graciela flora. But at the time, it was still called a Leda back when it was originally produced. And it's at least the one that's in the widest circulation that we've pumped a lot of into the market over the years, taking cuttings vegetatively. Always stays vegetatively, never fades from that. Holds a really nice variegation. It's a male. We've bred with it, and no variegated offspring. Sometimes it skips a generation with other plants. Obviously, the females that we've bred it with have not been variegated, so maybe 2 variegates might produce some variegated stuff. But we know with some certainty that it's not like even 10% of its offspring are variegated. So that's another one that's just mystery. We don't know which variegation it is. Its stability over decades does kind of imply genetic, but we don't even know where the gene for the sex of Nepenthes is, let alone like I think, unless something new has happened. So that whole genus is still a bit of a mystery genetically. So let alone like what causes variegation, we have no idea, and we don't have anyone even working on it, as far as I know. So it's kind of the Wild West, and a lot of it is just going to be empirical Mendelian kind of stuff, of literally like Drew and I swapping our pollen like between each other to maximize our ability to figure this stuff out. We still do stuff like that. And that's how we find out. But that's one I can shed a little bit of light that even a stable genetic variegation doesn't seem to do that much breeding wise right now.
I think the variegations in Nepenthes are probably not genetic. The reason I say that is, Drew, you spent a lot of time in the jungles looking at wild plants. I have. I don't know about Josh. I suspect so.
Nope.
And I have never ever. I don't ever recall seeing a variegated Nepenthes in the wild.
That's a super good point.
Yeah, no, that's a really good point.
I think they either just don't make it because they're not competitive, or they just don't appear. I've never seen even a young one. I really can't recall seeing one.
Yeah. Come to think of it, I don't think I ever saw one either. Seems a lot of weird mutations out there, but
yeah, they're very.
Like that. Yeah.
Drew, I was going to ask you. I have I had a 6-foot long gracilis that was like tricolor variegated. Then I hacked it up and took cuttings, and then the new basil, solid green. Why would it do that to me?
I feel for you, man. I feel for you. Yeah.
But it might, like Drew said, it really might. Yeah, like it might come back. Yeah, we've definitely, we've taken cuttings of our variegated Leda that we know always comes back, and I've looked at me like that is 100% green right now, and then leaves 6, 7, and 8 work their way into it over time. So it's not trying to give false hope, but we know so little about it, and even the people who know about it kind of just have to say like you'll have to just wait and just be sure to label them so that you don't like look at them and be like, why do I even have this, and then like chuck it and then find out that. Which we've done. So all
right, very good.
Definitely label things.
Yes, it is tricky because you do have those people, those collectors who are really keen on having them. And we do our very best to ensure that if we do sell, that it does have strong characteristics. But of course, people and it's stable. But of course, people do come back in years ahead and say, "Oh, my plant, isn't maintaining, and it's not stable." And it's a great shame because we do our very best to ensure that if we are selling something, that it is as stable as we can produce anyway.
We had an Indian gentleman came through here who collects variegated plants and nothing else of anything. And he just bought every variegated plant we have at that time. That was years ago, which means he cleaned us out, which is a pity because I would have liked to have done some experiments with taking cuttings and seeing if one side of the plant would maintain the variegation. I think it could be done.
Still take that payday though.
Yeah, well, you don't
Take
say
that
no
every
to something like that.
Take
He
that
had his
every
own private plane to take them back in.
Great.
So that was
Easy.
Oh,
Easy day. And then you take the week off after that.
no, now I do.
yeah.
All right, so we're almost out of time, but I want to, of course, ask Rob and Diana this question because we kind of started the whole conversation. What makes a unique cultivar, or in your mind, when are you ready to say, I'm going to name this, or I'm going to mass produce it?
Gosh, that's a huge open-ended question. Depends on who it's for. Unique cultivars, generally we've named them either because we want to name it after an individual that we admire. We haven't named that many, or we've named it because we want to patent it in the States, which we have done, or take plant breeders' rights in Europe. It's got to have a name. And then the name has to be registered. We're very lax at doing that. So we actually have lots of cultivars that we could name, and we haven't done so.
Or as in the case with the Dutch, they take a selection of plants that we feel that they might be interested in. And then if they select something, then either they or us come up with a cultivar name. Yeah.
There's a question here which maybe Drew or Josh could answer. I'll just give you an example of a cultivar name which puzzles me. Is Predator, because is it a single clone? You've got Predator A, Predator B, or is it the cross that's been named Predator?
It's a Grex.
I'm not sure.
The
whole Grex is named Predator.
Yes, that's my understanding of that. And a lot of the ones from that era, like briggisana, Saber, like the famous old school set from the late 90s, early 2000s that everyone wanted, those are all Gredges that that have a lot of variation. And then certain ones have landed in the right place to kind of become the flag bearer. So like with Predator in particular, like there's the Jeremiah Harris clone, where Jeremiah he ended up with one and everyone's got a cutting of that one. And I have several, and it grows really nicely. And it's a really nice representation of Predator. I don't know if any are named. There's several named Briggisanas that are actually registered. Even of Saber, there's like the Saber Dark Cherry, I think is what that one was. That's kind of like agreed in the community to be the best representative of that cross. But yeah, with Gredges, that's kind of how, I don't know. That's kind of how it organically ends up. Like I don't have it. I'm just like off the cuff. Like I'm trying to think of like what we've done.
Yeah, not a great
that
problem.
happens organically.
Maybe we should. Yeah.
But none of that, to my knowledge, is even registered or
well, it's
even
not official.
Acknowledged to be a Grex.
So it's not official, actually, from what I recall, right? And this is, I think Jan Schaller broke it down one time at the, when we were talking about this at one of the ICPS conferences, I believe orchids are the only family of plants that have officially adopted a Grex system. So officially, the names of orchids, right? So I don't know, like, I don't know, what's a good example? Paphilopetalum, micranthum crossed with the Paphilopetalum, what's the Delanti is Paphilopetalum magic lantern. That is accepted, like technically, right? But other plants, like if you wanted to do that with Nepenthes, technically that's not kosher. Right, like
there's
nobody.
There's no body regulating.
Yeah, there's no body regulating this, right? No, I mean that's a technicality, right? Like, am I all for giving plants a name that like doesn't intimidate beginners with algebra, like algebraic equations? Yes, I am. Right. So I think I do think that's the direction we have to head. I think that it's already been done with like Rob and Diana have already, Diana, right? That's a lovely cultivar, right? It's much less intimidating than say Ventricoca sibianensis ampullare to somebody.
Right.
I just named that one to make sure I got fed. But basically, a good example that springs to mind is the Babigia edwardsiana that we all use for breeding. It's all of one clone, as far as I'm aware. And we're crossing this with Babigia edwardsiana, that with Babigia edwardsiana. I think it might be a nice idea to actually name that. Sure. And just simplify the Latin and all the brackets that we put around things,
Yeah. So, and someone, but like Drew said, go ahead, because there's no one else doing it. Like you don't have to ask permission because unless like so famously, like Drew probably remembers this era back on the forums before Facebook. Sam Estes tried this very famously and very unsuccessfully in the end, because he got a lot of resistance from the community and it just didn't really take off. But there was he built a whole registration website where you would submit the mature characteristics of each given parent, and you would talk about how they grow, and you just like document all of that, and then you name your Grex, and there's like a Grex registry. And we were not doing our breeding program at that time, but I followed it closely because I was just kind of interested. And, you know, to give credit where credit's due, a lot of his decision-making and arguments for the necessity of this and kind of pontification about how it should work did inform the way that I designed my internal Grex system. So we have our own Grex system that we use for naming our hybrids, because again, it just becomes a big bracket nightmare, and most customers don't actually care what it is. They want to know about what the plant looks like. They don't really need to know what Grossola flora by Edwardsiana Burbigiae by what, like they don't need to like try and figure that out. You just show them either a picture of what the plants look like, or at least what the parents look like next to each other, because there is variation within that, right? So we built that program. I mean, essentially what brought his thing down basically was that breeders didn't necessarily want to, I think, silo underneath. Like he had as little authority as any of us do to set that up, right? He's not a naming body. He has no registration criteria. Like he built a thing and said, everyone come in, and it didn't really happen. But we did that for our own plants, and we're not trying to enforce that on anyone. Anyone's welcome to emulate what we did, but it's mostly to keep our plants straight internally, and so that our employees don't have to be doing math in the greenhouse, and so our customers can be collecting that kind of thing. So we did kind of build a mini unofficial Grex system with no pretenses of altering the way other people do their naming, other than showing them that it's pretty convenient and it works well, and we happen to like it. And you know, there's people who like it and people who don't in the marketplace. But it ends up really simplifying things on our end, and making it much easier and much less intimidating for people to become consumers of them, right? Like
Can someone confirm that briggisana, Peter D'Amato, and lowii times ventricoca red, they're all the same parents, but are we saying those are three different selections from?
No, specifically briggisana.
Or is it switched?
Well, yeah, but so briggisana was technically a specific crossing. It was a moment. It was like this lowii and this ventricoca. Sorry, I was getting confused between that and Trusmediensis. So whichever parent plants cross that, that's the Grex of briggisana. Because there's no regulatory body, there isn't actually a correct answer to one of your questions. The other one is Peter D'Amato is a selection of that briggisana cross. It's one of the actual selections. So it's a subset of that briggisana cross. The question comes up of, okay, so is now every lowii by ventricoca a briggisana? And like Drew said, in the orchid world, yes, because it's registered. And anytime you cross species A and species B, that is a Grex, and it's already named and it's taken care of, and we know what the characteristics are and all that. With briggisana, because it's not regulated, like there isn't actually an answer. Like, is every lowii? That one's not even briggisana. Like it's not. There's no one who says that that's briggisana, other than someone actually just put an X and said briggisana when they made it, but that's not registered anywhere. Peter D'Amato is registered as a selection of the briggisana cross, but future people who make that cross are, one, not obliged to call it briggisana, and two, potentially can just call it briggisana, because no one can say whether they can or not, because no one even said that was okay in the first place. So
yeah,
that's
Josh,
the gray area that we're in.
Yeah,
we
Josh,
name.
I'm a big fan of what Sam Estes was trying to do, but I
Right.
can see it from both sides. Because, for example, if we suddenly rush to name all the crosses we've made and all the you know, have all these Grex names, we suddenly came out with hundreds of different Grex names, and everyone was forced to use them. There'd be a lot of confusion as to what was actually in the plant.
Sure.
By the time we finished, there'll be all sorts of, and also people can name something, who was first. You know, it has to be properly registered. It has to be somehow controlled. Otherwise, it would just become a complete free-for-all. Which is why we've been quite slow about naming things unless they're done as a specific cultivar and properly registered through the ICPS. We did it, I think, a few times. But there are certain things we would like to name because it's just simple. It would simplify the nomenclature, but providing everybody else followed it. But if everybody else named them something else or didn't name them, it would become an even more complex quagmire than it already is on the nomenclature.
Right.
Rob and Diana, is Lady Luck and Bloody Mary the same plant?
Yes. I wish it was. I wish we had.
Singapore
Bloody
GardenTech?
Mary. We didn't like the name Bloody Mary, but then
It's not
it was
us. It's one of our
supposed to be,
shareholders.
yeah.
One of our shareholders was quite offended by the name
Yes,
Bloody Mary,
yes,
so
as
he
was
wanted
I.
us to change it. It had already been registered as as Bloody Mary, and he liked the name Lady Luck, and he was a pretty big shareholder at the time. He still is. So unfortunately, it became very confusing. We should have just stuck with Bloody Mary. That's what it's registered as a cultivar in Europe, and it's got plant breeders' rights, and also it's Bloody Mary in the United States, where it's got a patent. But it's I think in both cases it's also put AKA Lady Luck.
Yeah.
So it can be either.
Yeah, so we tend to name things specifically to maintain our ability to show their genetic makeup in the future if they do get produced by other people without our say so. So, and that has happened, and legally they are bound.
Commercial rights. Yeah.
Commercial rights. Yeah.
Yeah. Was Singapore GardenTech back, because this is old school kind of questions? What, that was a different selection of the same cross?
You're going really back. You're going to be
Oh
talking
my
about
goodness me!
All the way back to Singapore GardenTech. I got the first set of those. Tell me about that.
No, that was unofficial and can be disregarded, as can another one called Casling Goo.
Oh yes,
Casling Goo won a lottery and had a plant named after on the spur of the moment, which was never registered. So GardenTech was something we were sort of compelled to do to keep a politician happy for political reasons at that particular moment. I'm amazed anybody even remembers the name. So I
remember everything.
You seem to. You're a scary guy.
You can't get away with anything.
Yeah. I have one more question regarding names, and I'm blanking on it. But what is Viking? Mirabilis variety?
Oh, that is that's
Drew.
Complicated.
Yeah.
All right.
Taxonomy.
Drew's got this one.
I have a love for Thai species. Just very, very good friends with some growers over there who are doing a lot of conservation. So Mirabilis. Let's see. Well, I mean, think we have to kind of. I don't want to paint with too broad of a brush, but it seems like culturally there's different norms. For depending on the place where things are grown, right? And in Thailand, many Thai growers classify things, or they look at things a bit more characteristics of the plant than necessarily like where it came from. So I think that's kind of how Viking started. Like it was an unnamed species, and essentially there's two populations of things that were called Viking. There's one around a university, so I'm told. Around, I think Trung is the location, and then there's another one. I believe it's on the Andaman Sea, that has a different form, I guess, of what was called Viking. And then later on, I think they were all called. Memory serves me right.
Wasn't it Mirabilis varglobosa?
Yeah, there were Mirabilis varglobosa for a while. They were both populations were regarded as Mirabilis varglobosa. And then they came back and they reclassified the one on the Andaman Sea, on the Andaman coast, to be orbiculata, but they left the one in around Trung as Mirabilis variglobosa. So I think that's where we stand right now. And there are some characteristics that distinguish the two populations, but then I'm also told by field observers that it can be a bit muddled as well. So
didn't one of the populations get destroyed
What's that?
Didn't one of them get destroyed in a tsunami, or is that not? Wasn't that globosa?
So the one by Trung, I am told, it's by the university, so it is somewhat preserved. Okay. Although I need to check with some of my friends over there to see exactly how well preserved it is. And then the one on the Andaman coast was a bit more in peril. Environmental things and also due to poaching.
Yeah, I heard that one of them is, as you know, Mirabilis is very common, but that one of them, this just triggered a memory, like got really, really close to being in trouble environmentally. And it was like, you mean the one that's like everywhere? Like, okay, so that plant does have a really interesting contemporary history of like bouncing around and what we call it and who grows it, and it hybridizes like crazy. So like the nomenclature is all messed up from those populations being kind of interchanged for a while.
Yeah, no, I still grow a bunch of Vikings that are breeder plants that they just have great characteristics. I don't know. I'm not going to call them orbiculata. I'm not going to call them Mirabilis variglobosa because I don't know. And, that would be stepping into the realm of, I don't like to base things just purely on the phenotype of the plant in cultivation. I think it's, things for cultivation are best left for cultivation, and, horticulture and, taxonomic concerns are best left for, like seed with actual provenance, right? Like in conservation programs. So yeah, my Vikings are still breeders, right? They just have really cool characteristics, and I still call them Viking.
You were saying that you find that generally people are not really interested to have a specific names for plants. I mean, what their family background, the female, male, whatever background is, whether it's a compound hybrid or just a simple hybrid. But I find that Robert does go into a lot of depth when he's describing and sending out bulletins. What do you feel?
So it's audience, right? So I am Rob's customer, right? So when no, we're like that. Like, let's be very clear. When he's making those bulletins,
I get those bulletins.
To me, right? Me and Drew get those. So he's telling the growers who purchase his genetics about the origin of those genetics, and we are deep field experts for whom that information matters, and we can make decisions based on that for what we stock in our growing program to get into the market. Asking end customers to have that level of expertise and care, frankly, about what the implications are of it only being a quarter lowii versus half lowii, which is that it will probably have lid hairs versus probably won't have lid hair. Like, all of that is, I think, somewhat of a detriment to the marketing of the plant. That we need to talk about the features of this plant that I'm selling you, not of its genetic potential from the breeder who's telling me about what leads to that. So that's where I come out when I'm talking about. Plenty of my customers do care about the provenance and the Latin name and what subvariety of that Latin name. Like, totally, those people exist. But if a plant is like a great example is the only plants available at Home Depot, right? It's what we call Aleda and Miranda, and no one actually knows what those are. We speculate what they are, but that's not public information. That was never published because culturally, the firm that made those, and like, it's none of your business how we made those. And in fact, it's our intellectual property, what this is. We're not telling you anything about this. Like, yeah, we've made a story that Miranda is Maxima northiana maxima. Someone had some information and speculated and whatever, but it's a very weird selection of that plant. And it doesn't really matter what the parents are when it's just this one clone that makes these gigantic banana leaves and these huge pitchers. Like, its siblings almost certainly didn't do that. And so really the genetics matter up to a certain link in the chain, and then of the getting from you guys to the eventual customer. So it does matter along there, and it matters on some weird branches where people really want to know, like, is this, is this the globosa variety, or is this just Viking, or is this, like, what is the parent? Versus like, this is a really nice plant with really nice color and some teeth on it, and it grows in this way, and it fits really well on a windowsill, and it doesn't get too leggy. Like, that's more what we're talking about when I say that, like, and for those, you just give it a nice name. That's what, I doubt a majority of your customers know what Lady Luck is. We can all rattle off that it's Ampullaria ventricosa, but that doesn't matter to the tens of thousands of people a year who buy it internationally, because it's in growers programs who did grow it with the knowledge that, okay, it's Ampullaria, it needs to be pretty warm, pretty humid, but that means that it won't get root rot. It makes, bigger roots than most. So they know they do all of that math when you say with the crosses, and customers can't even pronounce Ampullaria. Like, why would you want that involved in the name when you've got something as good as Lady Luck, which everyone remembers? Like, that's branding.
And I would say this too, like, say you go to a big like show for like actually like commercially produced plants, like TPIE in Florida, right? Like, I went there this year, right? Like, I don't think I saw any species names of anything. There's no Latin there. Like, you look at other industries that are, frankly far ahead of carnivorous plants, and they've already gone that route. Like, you look at a whole catalog of anthuriums, it is all, nice sounding names, right? There's some marketing person who thought that had a nice ring to it, right? And same with philodendrons and, in other things, right, that are mass-produced tropical plants.
Yeah, it's very interesting.
So that begs the question: should we just be naming everything?
Yeah, with the Dutch Bloody Mary, I've been trying to get the Dutch to actually wean themselves off Bloody Mary onto Diana, because I think Diana is quite a similar looking plant, and it's got some superior characteristics to Bloody Mary. But there's a huge amount of resistance, because the name Bloody Mary is known by the market and known by all their buyers and known in the auctions in Holland, and they don't want to change, even though Diana may be a superior version of it. They won't change because that name is stuck, and it's been known for 25 or 20 years or something, whenever.
Well, they do say that their customers, it's red. So their customers don't worry about whether the picture's a bit bigger or a bit, that it's a red picture. So
yeah, there's only room in the market. Like, that's the other reality of Nepenthes, right? Like, being this deep inside of it as we are, we see all the subtle differences between crosses and this. Realistically, like, when you look at like much bigger markets like alocasia or colocasia, even those have a couple dozen varieties that have any representation in any international markets. Like, how many mass-produced Nepenthes can the market bear, right? We definitely know we already have like Miranda and the Aleda. We have like Rebecca Soper kind of floats around, and now we have Gaia, Lady Luck. Like, we have like 5 that are in production, in culture, being, churned, like bounced around internationally to hitting all these marketplaces. Can we double that? Like, there's not 100 Nepenthes that the market is going to hold. And so I do see their argument, like, yeah, that is a very nice red Ampullaria ventricosa hybrid, but like, we have one of the, like, we spun that one up. Yeah. We branded it. Like, that's, we patented that. We're trying to recoup. So I'm becoming more and more like, when I look at my stuff that I'm producing, I'm like, oh, that's really nice. But like, could someone actually be expected to tell the difference between that and Miranda? Like, it's just a big maxima. Like, is that something that an end user is going to buy when, like, a Florida nursery is cranking out 250,000 of those a year? Probably not. They're like, they're selling them a huge plant for like $50. Like, so they have the economy of scale, they have the first mover advantage. And so as the market develops, anything that I try to get into the market has to be clearly differentiated from every other plant that these growers might be growing, or I have to be cheaper, which is definitely not going to happen because I charge a huge amount for my plants. and or
you're in the Bay Area.
But I'm growing these in San Francisco, you guys. Come on, like, this stuff is expensive. And so those are the considerations that I'm now like that's kind of dawning on me, that even like lifelong, like kind of backyard breeders who are even in the industry, like they never have to deal with that, because you can just, if you don't produce that much, you have your whole market right there. You can go your whole life without ever worrying that this, complex Tivii cross looks a lot like that one I did 5 years ago. And like, it doesn't matter, because you just make your plants and sell them. If you want the general market to absorb your stuff, it's got to be something pretty special. And like I said, we're testing 500 crosses. A lot of those are just going to be dismissed offhand as like, I'm not going to try, like we just realized I spent a bunch of time making a bunch of pure red ventricosa. Like, this is just internally, because that used to be available in America, and then the culture just kind of went away. And so now you can't get ventricosa. It used to be very, very common, and it was several facilities were producing it quite a bit a year. And so it's just around, and then it's not. And I thought that was kind of sad. So I started focusing on replicating some of our in-house red ventricosa. And then I found out why it's not around. And it's because no one's going to grow it, because pure ventricosa is just in every measurable way not as good for growers as the D. rus alata. And so, and a customer cannot tell the difference between a D. rus and alata. That's, we're like, oh, but it's elongated and it has a different leaf attachment, and the peristome isn't as scalp. It's a kind of red plant that has an hourglass.
It's not a later.
Yeah.
It's not
It's
a
not,
later.
and it's also not
Okay.
but whatever
Basically.
that's what they sell it as.
It's not ventricosa either. Yeah, they
Yeah,
always
but it
have
doesn't
them,
matter,
but yeah.
because it's close
No, it
enough
doesn't
to
matter.
that to lock a lot of plants out of
Yeah.
the market. So then you have to do something that has some stripes or speckles. Okay, but don't get too close to Miranda or Gaia, because both of those already have it. So there's like this limited palette of traits that everyone's trying to niche into if they want to hit a real market that has any scale whatsoever. And so that's where the like idiosyncrasies of everyone's own breeding program and the unique plants that we happen to have, or the unique crosses that we've made, that's the only way to get into the market. Like, you just have to have something that truly is different. And it's not necessarily the toothiest, it's not necessarily the biggest, but it's something that looks different than what's out there.
And it's got to be easy and fast
That's
to
right.
grow.
Yeah,
We
You
spent,
know,
it has to go really well in Florida
quite a
or
bit
Southern California.
Yeah.
Yeah.
We spent quite a bit of time in the Netherlands last year with big nurseries, and asking what they wanted. And they're not interested in Latin names. They're not interested in anything other than 5 or 6 different characteristics, and it has to be different from anything that anybody else is doing at the moment. That's all they want to know about. The use of Latin names and the what we do, basically, we have to remember we're all nerds. As far as they're concerned, we're the breeders, and this is what we love doing, and we're into it. But the big new growers, like the Dutch, the end users are for our products from the lab, for example, in the new lab we're building, couldn't give a hoot what the parents are. And Diana's got a different parentage to Bloody Mary, aka Lady Luck. It's a different species parentage. They don't care. They just want to know if it's better or not, and is it worth changing the name on the market for something that does look fairly similar? And the answer was no, generally.
All right.
So we've wasted our time on that.
We'll get there.
It's the friends you made along the way.
All right. With that,
yeah.
I have one last question.
I still saw a lot on Diana.
Yeah.
You did a lovely,
Drew,
write-up. Drew, if you come and visit us again, Drew, you'll get a lovely dinner for writing that one up so beautifully. Every time you Google Diana, I come up with your write-up. Yeah, the lovely Diana. Yeah.
All right, so I'm hoping we can do one quick fire round. Could we just go around and say one cultivar or cross that you did not create, that somebody else created, that you admire, or something that you appreciate? Diana, you can go last.
Thank you for that.
Drew, do you have a cross that you...
Yeah. Okay. So this one, I don't think people appreciate what a horticultural achievement this is. Raja crossed with Rob Cantley. Those are like two, the dream cross, right? But see, I know what went into that. A lot of plants from that kind of Bornean clade, the ultramorphic ones like Mira, Raja, those ones are they can have some compatibility issues with some of the Philippine plants, right? So Rob, I think you told me a story where you got like 1 or 2 viable seeds out of an entire stalk on that cross. And then we're able to multiply that up over years and years to finally get into production. Yeah, no, that is, definitely a horticultural achievement right there to get those 2 giants. And there's still not mature plants out there, but that is something that I think is really admirable.
All right, Josh.
Yeah. So like we're both talking about Rob, but he's got quite a bit of time on us. So it probably will always come back to something that he made. But favorite hybrid in the world is Mulensis by lowii. Best plant that I'm aware of. Everything I love. I have my own that it was I think it was in my first order from Rob in 2009. If not one of the ones shortly after that. But every time I run across that cross, I'm just like, I wish I could do, like, I've never made one that was quite like that really was the plant that I wanted to grow more than that one. And so that's the one that I would always like trade for, was like the Mulensis lowii. And that's from what I remember, that might have even been like a collection, like an opportunistic one. From
Diana, that was a really easy one to come by, right?
It's just around, or is that? Yeah, but that one is my, like, just when I'm talking about like what I like in a hybrid, like Mulensis lowii, like that's the plant that I like to grow, and I would go to bat with that one.
Well, I've got a little story that when my mother came out, we were talking about the rainforest and the natural habitat, and Mum said, well, somebody must have planted them. And I was just getting to the end of my tether, and I just said to Mum, yes, God. But and on that note, Alice Putrana is a beautiful natural hybrid, I believe.
Yes.
Yep.
Yeah. We duplicated it, but to burst Josh's bubble, ever so sorry, but we didn't make Mulensis lowii. It was opportunistic. It came
that's right. I remember that. Yeah.
But I think I admire a lot of the breeding that's going on at the moment. People that have species that we don't have, particularly Edwardsiana crosses. There's Jeff Schaefer's doing fantastic work. In fact, a lot of other people too. Jeremiah breeding stuff that's not yet been named, but there's all these Edwardsiana crosses coming out, which I think are going to be fantastic, but we yet to see them. But Mother Nature, in my opinion, produces some of the best hybrids of all. Some of them we're using in breeding programs today, because she's had quite a lot of time on her hands to actually get it right, and they've always got a reason to exist.
Mulensis by lowii.
Yeah, exactly. I love natural hybrids. It's a personal thing of mine. Whenever I was in the wild, if I could find a hybrid between the species I was looking at, I was always really happy. But yeah, I can't give you a specific name. I wasn't really prepared to think of one offhand, but the breeding programs that are going on, particularly in the US that I'm aware of, using species that we don't have, Edwardsiana is a classic one, are very admirable.
I have to also recognize that Jeff Mansell in Australia has done some really nice work with a lot of plants that are a bit warmer growing than have been developed in a lot of other nurseries, and I think that has creeped into a lot of breeding programs. I know I use some of that stuff as a backbone of some of my crosses, particularly with Veitchii and Truncata hybrids, and I think a lot of those plants are well suited. They're very popular in the U.S. because the U.S. is a bit warmer than many other climates. So I mean it's often your breeding can be influenced by the climate you're developing those crosses in.
Right.
All right.
Yeah, you definitely flower very different things than I flower, Drew.
Mm-hmm.
Or like our greenhouses are quite different. Like San Francisco and Austin have very different climates, and so we are not regularly flowering the same stuff, except at very different, like very specific moments when our climates maybe cross over a little bit. And so, yeah, my program is very much a product of living on the coast of California, for being able, like I've flowered truncatas and tentaculatas and just weird stuff that had never been used before. mostly because most people's climates are not quite the same as, 3,000 meters in Borneo. Mine happens to be. And so I've made a lot of crosses that probably only do well here and definitely will not succeed in cultivation, but they're kind of cool and they're probably not going to happen again unless someone in my area is doing this. So
yeah. No, I've noticed, certain growers are able to cross things regularly that I can't get to flower at the same time. I think it's just it's purely a climate thing. I've got some tricks I'm working on right now for that. Right, like we've got actually an interior grow room that's 100% artificial inside, and so I'm messing around with reverse photoperiods and things like that to mix some things up, and we'll see if it works. I don't know. Maybe I'm trying to be too smart about it, and it's just going to fly in my face, and they're going to still flower at the same time, right? Due to something I'm not really monitoring.
Yep.
All right. Thank you so much, panel, for sharing your expertise on breeding Nepenthes, and thank you for trying and failing and then trying and succeeding. And more importantly, sharing with the ICPS members.
Mm-hmm.
All right. Thank you, everyone, and have a great day.
Thank you.
Been a pleasure.
Really enjoyed it. Thank you.
Nice to see you guys.
It's
Bye,
not
everyone.
a surprise
Bye.
that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.